I work on a group of proteins called protein kinases, and I use structure of the protein to try to understand function of these proteins. And when I started working on the protein kinases, there were only a few that were known. And we subsequently learned the sequence, and then because I was trained in a crystallography protein chemistry lab, I focused on getting a structure of it, a crystal structure, so you could see the molecule in 3 dimensions. And so that was a very major focus, and we solved the first protein kinase structure back in 1991, and by then it was getting to be a large family. Now it's over 500 proteins, so it's one of the largest gene families, and they regulate all of biology almost, and there are many that are associated with disease, so a dysfunctional kinase can lead to a variety of diseases, especially cancers. You have one mutation that the kinases are switches, and so they get turned on and off, and if you have a single mutation that can keep it constitutively on all the time, then that can often drive a cancer, and so they become major targets for drug discovery, and our structures have really provided a foundation for thinking not only how our structure looks of our one kinase, but it's been a prototype for the entire family, and thinking mechanistically about how they convert from an inactive enzyme to an active enzyme has been revealing to a broad range of biology. And so trying to see how our kinase structures and information about the kinase can lead to an enhanced understanding of biology and disease is kind of a driving theme. I was very pleased and honored to receive this award named after Thressa and Earl Stadtman. They were close friends of mine. Earl Stadtman, I never worked directly for Earl, but I came to be a good friend. He was always an advocate for me, and whenever I visited NIH, when I would come like first on study sections or later on the board of scientific counselors for the Heart and Lung Institute and on GM Council, every time I would stop and see Boone Talk and Earl Stadtman, and sometimes I'd give a seminar, sometimes I wouldn't. We'd just talk science, and we'd go for dinner afterwards and have a nice glass of wine. And so over the years, I unfolded my story to him, and he always was so appreciative. And he and Theresa, they're pioneers of biochemistry of the last century, and so I'm very deeply honored to receive this award. I was a past president of the society, so I'm very dedicated to the society, and one of the things that we've always wanted to do is make it an exciting place for young people to be and for people to share their science. And this meeting, I think there are more people attending this meeting than in a long time. It's a lot of attendance, a lot of students here. I've really enjoyed talking with students and postdoctoral fellows, and a lot of people have come up through the last couple of days after my talk and commented on specific ideas and how to follow through on things. So I've really had very, very interesting scientific discussions with a lot of people, and I'm very happy to see kind of the excitement that a lot of young people here, a lot of people at the posters, a lot of crosstalk, and I think that's what we strive to achieve, and hopefully we'll continue. It's an exciting meeting. I think it's more exciting than in a long time. We have more people here, and a lot of crosstalk, a lot of enthusiasm at the sessions.